So this is the second part of the Cells and Environment lecture. And here I'm going to talk about the dynamic cytoskeleton. So we've already shown you told you about the cytoskeleton and how it sort of helps form the structure of cells, but you shouldn't think of the components of the cytoskeleton as being rigid. So in fact, the cytoskeleton is constantly reorganizing and it's not static at all, and it's constantly moving. So I'm just going to start this video here so you can see it, and this shows us the actin filaments, and here are the actin filaments, and hope you can see in the red color at the edge of the cell they're constantly extending and contracting. They push out the plasma membrane of the cell as the cell senses its environment, perhaps for nutrients, perhaps to find a neighboring cell. And when cells move, like this cell was, hopefully you can see in the video that the cell is moving in this direction. You can see that the actin network can actually be remodeled incredibly rapidly to enable that movement. We'll talk more about cell movement again. Another example of the cytoplasm being the cytoskeleton being dynamic are the microtubules. So you can see that the microtubules remodel to allow this to allow the cell to send organelles or vesicles to new parts of the cell. In fact, you can see an example of essentially the walking along here. Again, that involves intermediate filaments connecting to the microtubule network to move vesicles about, and it's also very important during cell division. So let's talk about cell division in a little bit more detail. So what we're seeing in these pictures here is a cell under a scanning electron microscope, and we can see the cleavage furrow here, which is the beginning of the cell separating into two new daughter cells. And what happens in that cleavage furrow, as they're beginning to separate, we get a contractile ring of actin filaments, and hopefully you can see it a little bit better over in this diagram here, where that ring is that center structure, which is green, and it is connected to the microtubules, which are around. So if you're colorblind, I'm sorry, they both appear brown, but I've tried to highlight those two structures. And again, we can see that is the actin there, and this region here is the microtubules. So you can see the interaction of two different components of the cytoskeleton in that division from one cell into two daughter cells. So how does that happen? And the process of this dynamic approach is a process of polymerization and indeed depolymerization. So let's start off first of all thinking about the actin filaments. Okay, so we've already mentioned they grow at the plus end and they shrink at the minus end. And this is what we call treadmilling. So at the plus end, we're getting new things added at the plus end, and at the minus end, they come apart and then get added again at the plus end. So it's almost like a treadmill. Microtubules, as you've probably already seen in the video, they both grow and shrink up at the plus end. Okay, so the minus end is normally anchored to that microtubule organizing center, which is near the center of the cell. Okay, so when the cells are growing, then the two monomers, alpha tubulin and beta tubulin, join together and add and start forming to the plus end. During the shrinking phase, those dimers start to unwrap and pop off. Okay, so this is what we call dynamic instability in terms of microtubules. And in order for them to remain stable, they actually need to be capped to prevent them from going into the shrinking process. And indeed, they shrink at about 1,000 times faster than they grow. And again, that's about 100 times faster than they grow, and that's dynamic instability. So they do need to have that cap put on at the end. The intermediate filaments are slightly different. You begin with the tetramer, and then like the protofilaments, and then they polymerize together to join larger filaments. So it's kind of like a unit construction that gradually gets larger. And again, they're subject to depolymerization, and these filaments can be broken down. So what roles does this play in actual cell behavior? Well, the first one we're going to talk about is cell migration. And cell migration is really important in lots of biological processes. It's really important in the spread of cancer, so that's a pathological example. But, really useful things like forming new blood vessels, wound repair, development of embryos, and your immune tissues, which the cells in your immune tissue will move to the sites of injury or infection. And during this migration cycle, there tends to be several phases in how the cell moves. So if you can see this video, we tend to get protrusion. It's where the actin filaments push their way out, kind of move into a new environment. They then tend to stick to whatever they are moving into, and then they kind of pull themselves along, and that's called protrusion, adhesion, and then the trailing end of the cell is then retracted. So what we can see here is that here the cell is protruding out, and then it's adhering to the new surface, and then we tend to get retraction here, and eventually this will be pulled back into the cell body. And this process, you'll be unsurprised to know, requires reorganization of the cytoskeleton. So let's first look at the role of actin. And it's important to remember that I've already mentioned this, they're rigid, they provide structural integrity to the cell, and they are much more rigid when compared to the plasma membrane, so which is very elastic and fluid. So if actin filaments grow at the front end of the cell, then they will push the plasma membrane forward. And hopefully you can see that on this video, that the green color we see in that video is the actin, and as that protrudes forward, the plasma membrane sort of follows, is pushed forward with it. The membrane is then attaching to the surface where we get adhesion and the cell is pulling forward. Now, the retraction process is that we have a finite pool of actin within that cell. So as we're moving from left to right in that video, as those actin filaments are moving forward, they're also breaking down at this trailing edge here and then being shuttled forward. So that's the retraction phase. And it's called treadmilling. And you can see treadmilling in action in this little GIF down here. And it's almost like wiggling forward because it's expanding at one end and retracting at the other end. So another important idea of cell migration is what we call chemotaxis. So chemo is a chemical and taxis is movement. So here what we're talking about is cells sensing the environment. So again, the actin filaments primarily here and the cytoskeleton allow rapid changes in shape, direction in response to signals from the environment. So, for example, we have here a leukocyte moving towards a chemical nutrient source. We can see in this video the cells, the little dots that appear are nutrients where the chemical attracting them is being released. And you can see it in a more dynamic process in this video where this kind of shady bit here is a pipette filled with a chemical which is attracting these cells, and the cells will move towards that chemical. So here you can see an example of a cell with most of its actin at this end, but it's getting a signal here at this end. That could be a nutrient source or enabling cell. So what happens is the filaments disassemble and they start to assemble in the direction of the nutrient source and move towards it. Now, as well as allowing cell movement, this dynamic ability of the cytoskeleton to change shape allows cells to have different shapes. And you'll see that different cells that have different functions can have rather different cell shapes. So a red blood cell, due to the organization of its actin skeleton, looks very different to a nerve cell. And you've got to remember that the nerve cell can actually reorganize, form new connections to strengthen or weaken certain neural pathways. So hopefully that's quite obvious, but the change in the shape of these cells is largely down to the cytoskeleton organization. This is a really neat little picture on the right hand side where you can actually control that to some extent in the laboratory and make a cell grow in specific shapes based on the environment the cell is put into. We'll stop there before we go on to the next section.